Good day, everyone, or good evening from my home country, the Philippines. My presentation is all about my independent research entitled A Verified Checklist, Lectotypification and Description of New Species of Begonia in Mindanao, Philippines. My name is Mark Arcebal K. Naive. I am a faculty member of the Jose Rizal Memorial State University, Tampilisan campus, and is currently pursuing my doctoral degree in Xishuangbanna Tropical Botanical Garden of the Chinese Academy of Sciences. So just to give you a short background about my home country, the Philippines, it is a country of Southeast Asia, consists of more than 7,100 islands strewn over the Western Pacific Ocean and is divided into three major islands. We have the Luzon, Visayas, and Mindanao, and harbors over 5% of the entire world's flora, thus considered as one of the world's megadiversity country. It is one of the planet's most geological complex tropical area, thus considered as one of the Center for Biological Evolution Studies. And unfortunately, the Philippines is among the world's 25 top biological hotspots, which ranks second. Therefore, these species are under extremely high threats for extinction. Just to give you an overview as to the extent of forest cover loss in the last 100 years, so from 70% forest cover in 1900 down to 18.3% in 1999. So imagine how many species have gone extinct because of habitat loss and forest destruction. Without timely intervention, further degradation of our forests will continue and may eventually result in species extinction before the full exploration of their taxonomy and phylogeny, as well as their economic and ecological importance. One of the largest and highly endemic taxa in the Philippines, which are highly affected by habitat loss, is the genus Begonia. Begonia is the sixth largest genus in angiosperm and is represented by approximately 2,052 accepted species belonging to 70 recognized sections. They are distributed in the tropical and subtropical regions of the world, which are growing as terrestrial and or lianas, and these species are thriving in different types of habitat, such as in stream banks, deeply shaded forest with the highest diversity in forest over limestone. The genus can be recognized by having these following characters: herbaceous habit, persistent stipules, asymmetrical leaves, monoecious flowers, inferior ovary, and three locular fruit. Economically, begonia species are primarily known as cultivated ornamental mainly because of their attractive leaves, which varies in shape, size, color, and variegation. Traditionally, they are used by the local people as a remedy to treat several illnesses and diseases and as a food, such as the flower infusion, which are reported to promote blood circulation and elimination of toxins from the body. Qualities of flowers are used to treat burns and toxic sores. The leaves are reported to treat cough and headaches. The leaves and flowers are also reported by the local people as edible, which is served together with other vegetable as salad. And the leaves of some Philippine endemic begonia species are reported by the local people as a storing agent for their local cuisines. Going back to the taxonomic history of the genus Begonia in the Philippines, the first taxonomic account was done by Merrill in 1912 with 59 species, and in 2008, the number doubled to 104 in the account of Mark Hugh on his an annotated checklist of Southeast Asian Begonia. Since then, the number of researches dealing on the genus Begonia have increased, especially the work of Dr. Robite, and is now represented by 149 species. Of the 149 species recorded, 97% of which are known to be endemic and is classified into two sections, namely section Barriandra and Petermania. Despite of the increase of researchers working on the genus Begonia, most of these work are actually focused on the island of Luzon of the northern Philippines, and Begonia species in Mindanao remain poorly known and understudied. As a matter of fact, Mindanao begonia are currently facing a lot of challenges taxonomically. Many of these species have not been recollected for over a century and is only known from the type material as well as protologs with an incomplete species description. Thus, most of these species are not yet assessed phylogenetically and their IUCN conservation status are not yet evaluated. One of the problem also is the existence of the unresolved species complexes, and there are still more species awaiting discovery and description. As part of our efforts to improve our understanding of Begonia in Mindanao and to facilitate biological studies for this genus more broadly, we aim to achieve these following objectives. First is to determine how many Begonia species present in the island of Mindanao, to redescribe and lectotypify 5 Begonia species, which are the Begonia affinis, Begonia colorata, Begonia malindangensis, Begonia mindanaensis, and Begonia oblongata, to conduct preliminary conservation assessment for these 5 endemic species, and to provide a complete taxonomic treatment to the interesting unknown Begonia species collected in the province of Bukidnon. This study hopes and aims to provide a complete revision of the genus Begonia in Mindanao, which will serve as a supplementary information on Philippine flora and a contribution to other related studies, such as in phylogenetics, biogeographic, and pharmacological studies. This study also serve as a baseline information essential for the sustainability and conservation of these highly endemic taxa. Moving on to the materials and methods, so prior to the conduct of this study, gratuitous permits were obtained in the different localities where botanical excursions were done, which is at present have only been done in the provinces of Bukidnon and Zamboanga del Norte. We also have examined the existing collections of the different herbaria and have conducted an extensive literature review. Morphological analysis was done both in the field and in the laboratory, and the collected specimens were prepared as our voucher specimens and are deposited in the different herbaria. Leaves were preserved in silica gel for molecular phylogenetic study, and the flowers were preserved in 70% ethyl alcohol and were subjected to stereomicroscopy, allowing us to provide a more complete and detailed species description. The species were then identified using relevant literatures and databases. As for its conservation assessment, the distribution information of each species were gathered from its herbarium specimen data and based on our recent fieldwork. These information were then used in the calculation of its area of occupancy and extent of occurrence in the GeoCat system, and the assessment was carried out following the criteria of the IUCN. And now for the result of our study, a combination of literature review, herbarium specimen examination, and recent fieldwork revealed that there are 39 Begonia species present in the island of Mindanao, which includes a new endemic species which will be discussed in the latter part of this presentation. Of the 39 species recorded, 38 are known to be Philippine endemic and 31 are Mindanao endemic. So the number of species constitutes 26% of the total number of Begonia in the Philippines, and these are divided into 2 sections. The section Bariandra with the least number of species, which is 9, and section Petermania with the highest number of species, which is 30. So these are the different Begonia species present in the island of Mindanao, of which 6 will be discussed shortly. Through this study, we were able to recollect Begonia malindangensis and Begonia oblongata, as well as rediscovered Begonia affinis, Begonia colorata, Begonia mindanaensis after a lapse of over a century. We have then provided a more complete morphological description and colored photographs to aid easy identification for each species. Our IUCN conservation assessment revealed that Begonia malindangensis is least concerned, Begonia oblongata and Begonia affinis as endangered, Begonia colorata as vulnerable, and Begonia mindanaensis as critically endangered. We also then took this opportunity to conduct lectotypification for each species, and these are the different lectotypes designated for each species. And now for the highlight of this research, we were able to discover a new endemic species which we named Begonia olganunezae, Naive and Kababan, published just this September in the journal Taiwania. The species was found to be closely similar to Begonia affinis and Begonia bangsamoro by having terminal inflorescences and staminate flowers with 4 tepals. This species was named in honor of Professor Dr. Olga M. Nuneza to recognize her efforts in documenting, studying, and conserving the flora and fauna of Mindanao while mentoring the next generation of scientists, including myself. As for its distribution, this species was so far Mindanao endemic and can only be found in the province of Bukidnon. As for its conservation status, we provisionally treated this species as endangered. Indeed, the Philippines is home to unique and highly endemic Begonia species. But the continuing habitat degradation and forest land conversion are major threats to Philippine biodiversity. These are attributed primarily to large-scale and indiscriminate logging and mining, burgeoning human population, over-harvesting of resources, and infrastructure development. If habitat degradation and land conversion are tolerated today, Filipinos will endure and suffer the loss of lives and biodiversity, displacement of indigenous people, and decline in agricultural productivity. In conclusion, the study highlights the first verified checklist of begonia species in Mindanao, which will help us refine estimates of true Philippine begonia diversity and provide us open access tools for accurate species identification. The study also highlights the rediscovery of two presumed extinct Mindanao endemic species, namely Begonia affinis and Begonia mindanaensis, thus enabling us to further clarify the controversy behind the identity of Begonia mindanaensis and have synonymized Begonia bolsteri to Begonia colorata. In addition, we have conducted lectotypification for the 5 Begonia species, which was done in accordance with the Shenzhen Code Article 9.3, 9.11, and 9.12. And lastly, the study highlights the discovery of Begonia olganunezae collected from the province of Bukidnon. Just to give you all an overview to what we have accomplished so far, so at present, we already have produced 4 papers through this project. 2 have already been published in Taiwania and in Phytotaxa. 1 have been accepted for publication in Q Bulletin, and another one was recently submitted in Phytotaxa and is currently undergoing peer review process. This study is still far from complete, thus we highly recommend to conduct wider sampling to improve species representation of Mindanao begonia, to conduct intensive field exploration of the flora of the Philippines, as there are still a lot of species awaiting discovery and description, engagement of the local people and young generation of scientists in exploring and conserving our Philippine biodiversity, and lastly, ex situ conservation for future molecular phylogenetics and pharmacological studies, and for sustainable conservation of the highly endemic begonia species. I would like to acknowledge the following for helping me in the realization of this project, especially to the International Association for Plant Taxonomy and Yayasan Conservasi Bayota Lahan Basa for the funding support. Before ending this presentation, I would like to share to you the quote of Carolus Linnaeus, which says, Nomina sinesis perit et cognito rerum, which is translated as, if you don't know the names, your knowledge gets lost. Thank you very much for your kind attention.